Okay, thanks everybody for coming today. So this is the second of our data science AI theme seminars, and yeah, so these are originally scheduled as quarterly. We had a bit of a break for the summer, and I think what we're thinking actually is they might become a bit more frequent. So maybe every 2 months we'll start having them. Today we're going to focus on UK Biobank. So Donald's going to talk first, and then Barbara's going to do a presentation as well. But the other thing is that, so maybe when during the session, if there's any other topics that you're keen on that might have as a seminar, then please do come speak to Hong-An or myself, or you can email us. A couple of ideas that we've got for future seminars. I think we'll probably get somebody from Open Safely in to talk about that data and how we can access it in the next couple of months. And one on the new TRE that's being set up. Those are just 2 ideas, but if you have any others, then yeah, just please come and speak to us. But I will hand over to Donald.
Hi everybody, how you doing? My name is Donald. I will talk to you about Biobank. I have a Biobank lanyard, which is how you know I speak with some authority. So the thing about this talk really is that Biobank is really an absurd resource. It is every type of variable you could possibly think of. And in that context, I will introduce it to you with a bias towards the stuff that I have tended to work on, in the same way that if someone else in a different field came and introduced it, they would have a slant towards their thing. As long as you know that's kind of the context, bear that in mind. And at the end, there's loads of resources that I've shared, papers and things that people can look at to really take their interest forward. And I'll talk a bit about what is Biobank good for and what is Biobank maybe a bit rubbish for. So what you have here, this is a Padlet. So I am not interested in reviews of my lecturing, but if you have questions, you can put them up there, and then we can ask them at the end if you feel a wee bit wary of sticking your hand up. And there's a QR code, so you can ask anything you'd like about Biobank or the price of cheese or what's the weather going to do later, and I'll try my best. So what we're going to talk about is about the basics of Biobank, kind of foundation, some of cognitive, the disease data fundamentally, because a lot of the disease data is kind of organized in a singular way. Some of the imaging, so that's like the heart, the liver, the brain, some mental health data, the typical kind of covariates that we tend to use, which will vary by study, obviously, some limitations to Biobank, and there are some quite meaningful ones, and then a little bit about the practicality of how do you actually get the data and use it. So very fundamentally, my interest is in predictors, mediators, and modifiers of cognitive health. There I am holding a copy of my own brain. And how do you go from, say, genetics to aspects of brain structure and function, and then cognitive health? And there's my brain scanned with the very fancy 7T brain scanner that we have in Queen Elizabeth. So what do I advise new users of Biobank? First of all, I think as per standing on the shoulders of giants, find a paper that is very broadly what you tend to do, broadly into your topic. You don't need to take photos of any of these. I'm going to share the slides afterwards, so don't worry about it. Then have a look at the data showcase, which we'll look at momentarily, to see if anything complements those studies that have maybe been done on your rough topic. And then I tend to tell students to come up with an analysis plan. This is kind of best practice in 2025 anyway, to go in with a clear method before you actually start with analysing the data. But also, I tend to find, particularly with student projects, it means that they can't go too off-piste with their intention. They can't say, oh, this wasn't statistically significant, I'm going to do it differently. So I find that a good way of doing it, and I do this myself. So Biobank, there is a degree of where you begin. It has data on anything you could really hope for nowadays, and that's health data, cognitive data, psychological, questionnaires about lifestyle, sleep, diet, smoking, alcohol, blah, 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 on and on and on. There is accelerometry data, which is essentially things put on people's wrists that they measure movement for a week or two. There is essentially just everything under the sun. So what this shows is the structure of it. And this is kind of the beginnings of one of the limitations of Biobank. That very fundamentally, in 2006, 2010, 500,000 people from across the UK, with a 5% response rate, attended one of 22 assessment centres. And they provided all sorts of things that could be reasonably analysed in a one-day assessment. And they also had blood samples and urine samples and every other kind of sample you could hope to collect, I guess, taken, and then that data was analysed over subsequent years and has derived things like blood biomarkers, urine biomarkers, all sorts. And subsequent to that, you have various reassessments which have continued to date. So 2011, there was a diet recall, so that's people saying what they'd eaten in the previous day. A repeat assessment, which is, in my opinion, a criminally underused kind of sub-cohort of 20,000 people who repeated the whole thing completely, verbatim, about 5 years later. Then there's accelerometry data, so that's things like sleep, physical activity. They did the cognitive function a bit better in 2014, and as you can see, there's just various subsequent reassessments that were administered to subsamples of the entire cohort. Nothing really is the full 500,000 again. All you've got is that first 500,000, and then maybe 200,000 people do one assessment, maybe 100,000 have done brain scanning. They've never really, that 500,000 assessment is kind of gone. They plan to reassess as many people as possible in the next few years, but obviously a number of people will have died or moved or, you know, shipped off to Portugal or all sorts of things. So very fundamentally, for my purposes at least, I'm interested in the fact that there is cognitive data at the baseline, there's brain imaging from about 2014 onwards, and there is linkage to NHS records, so hospital admissions and episodes throughout. So knowing that fundamental structure, how do you go about actually seeing what variables there are that are of interest to you? And a really good stop, I think, is the data showcase. And this is a very easy to use aspect of the website, which starts with essentially just drop-down categories. And it'll show you some of the data and some of the variables that they have. So what I'll now discuss is, I'll talk a bit about the disease data, a bit about cognitive data, mental health, imaging, and then genetics, which is not all of the data that there is in Biobank, but I think it's a half decent start. So very fundamentally, disease in Biobank is organised between self-report and or kind of participant completed, so there's sometimes questionnaires, and electronic health records. So a downside of the self-report is that obviously it's only completed when people actually did assessments, so it's dated to whenever they completed a questionnaire or the baseline, and so that means that you only really know that they reported that condition at that time point. On the other hand, there are NHS ICD-9 and 10 records, and they're increasingly supplementing that with various things like primary care data linkage, ONS death register linkage. They're only getting more and more linkages there, and increasingly Biobank are trying to have primary care in everybody. And they're coming up against it a little bit because GPs don't like to sign off primary care data. Biobank have very handily collated self-report and all the records that they have into variables called first occurrences. And what that means is that for this particular participant, for this particular condition, whether it's self-report or primary care or hospital admissions or whatever, this is the first point we are confident that they had this particular condition, whether it's heart disease or cancer or all sorts. This has not been done with everything, and there's a slight bias where only England at the moment has primary care data, which can be a bit of a collider, which means that that can it can matter that it's only in England. So, for example, recently someone looked at green space and blue space and how that correlated with mental health stuff, but it actually matters that that's only in England, which has slightly different characteristics to the rest of the Biobank. And very fundamentally, obviously, prevalence is anything to the point of baseline, and incidence is anything subsequent to baseline or whatever point you dictate. A downside of brain imaging, for example, I think, is that I personally don't think you can really look at incidence subsequent to brain imaging yet, because it's only really been a few years for the vast majority of them. Not that that stops them. So here's an example of the first occurrences stuff. So here you can see that it includes primary care, a hospital inpatient, death register, Hard to go far beyond death register data, you'd have thought, and self-reported data. So here, for example, I tend to look at dementia, and I often will look at first occurrences because it includes more data than just self-report or just hospital admission data or whatever. Biobank, it's not a Biobank document, but there's a good report by Kathy Sudlow from 2024 all about how the UK can leverage its population data better. And it includes in it a very interesting detail that Biobank know internally that they are missing tons of cases of various conditions by not having the primary care data. And that is why they're very, very keen to use primary care for everything, because they can see internally that if you didn't look at primary care data, you're missing swathes of cases of various conditions, which influences all the estimates that we're looking at. It's quite fundamental. So I'll give you an example of some papers of what we did. I'm not necessarily saying a particular list of papers. Like your children, you dislike each of them in their own ways. But I'll give you a little bit of kind of here's what we did using some of that data, and here's why we think it's vaguely. So here, for example, we were interested in we used baseline cognitive data, and we were interested in do people with better cognitive health baseline have a lower risk of dementia? The answer was yes. That's just a very basic, at this time point of something which was fixed to that date, do people have high risk of dementia? And does it come on earlier? The answer was yes. There we controlled for age at time of assessment. We only looked at people who were a certain age and above, sex, smoking history, deprivation, relatively standard stuff, I think. So I'll move on to the cognitive data a wee bit. I don't know if many people intend to look at that much. I think it's criminally underused, although I would. The Biobank data for cognition is a really good example of something that Biobank is an amazing resource, but they had a day to assess everyone at baseline. And that meant that the phenotyping maybe isn't as detailed as you would ideally like. And cognition's quite a good example where a proper cognitive assessment done by a neuropsychologist might take a day. They might do assessments like the Wechsler Adult Intelligence Scale or memory scales or all sorts of things, and it can take hours. Biobank, I think they're over in about 10 minutes, tops. There is a reaction time test, which was 4 trials, which took a moment. There's a quote-unquote intelligence test, which actually includes some questions which are not really what you would call fluid cognitive ability, but rather acquired knowledge. Essentially, they're just not they're not as precise or detailed as you would like if you were a neuropsychologist looking at data ordinarily. So here, for example, here's a minor limitation to the Biobank cognitive stuff, where the whole idea of fluid cognitive ability is meant to be that it's like reasoning. You can reason your way to the correct answer. Whereas what they have here is what's called crystallized cognitive ability, which is essentially just you know it or you don't, which is not really what you want in a test of this ilk. So there are certain little aspects to Biobank assessment which are novel to Biobank, and they're sometimes not perfect. However, subsequent to that baseline, they additionally added more cognitive tests, which are essentially legal knockoffs. So sort of, do you remember M&S and Colin the Caterpillar, all that? They're kind of knockoffs of proper tests. So it's not matrix reasoning, it's matrix completion. It's not digit symbol, it's symbol digit. Stuff like that, which are based on more traditional cognitive tests, which are very validated, but they are, for legal purposes, completely distinct. Can't make that clear enough. And these tend to be a lot better. They tend to be a lot more sensitive to age differences and to things like heart disease, diabetes. They tend to be more sensitive and precise, generally speaking. But they will never have that baseline sample again. So these things were all done subsequently to baseline in 100,000, 200,000 people years after baseline. So that's the slight downside of Biobank, is that 500,000 is gone forever, basically. Here is one of the early papers we did, and this was a very basic additive thing of multimorbidity and cognitive abilities. And it was literally just, as people have more cardiometabolic conditions, they have worse cognitive health. Easy. So these are quite good examples, I think, of if you want to check out some existing studies and what we did and some of the covariates we used, they're kind of a good jumping off point, I think. Mental health data. Mental health data in Biobank is really quite interesting because you have to look at several different things. You can't really, it's not really considered good practice to just look at one way of measuring depression in Biobank. So there are self-reports that are simple. I have depression, yes, no. There is history of depression. There are various detailed nuances to how depression is characterized in Biobank and when. The consensus is you have to use at least 2 to be characterizing depression in Biobank. So whether that's self-report and hospital admissions, and if you think about it, those are quite different things, really. So their first record of depression being when they're admitted to hospital is quite different from somebody going to see their GP, for example. So depression's quite a complicated one in Biobank. This paper, among quite a few, are very good for characterizing how should you best estimate depression in Biobank, if you want. And that's a whole big area is depression. That's really one of its main areas, I would say, Biobank. And here you can see, this is a really nice example, I think, of how you have to look at various different sources if you're looking at depression. So this is first occurrence of depressive episode, and you can see that there's a mixture of primary care, hospital admissions, self-report, and various things. A minor note from a paper that we did in 2016, though, is when we looked at low birth weight very early on in Biobank, we found that there was an association between low birth weight and high risk of depression using self-report. And this is before all the stuff that you should ideally use various different ascertainments. And what we actually found is that when you varied the type of ascertainment you use, it made absolutely no difference to the final estimates. So it's very important to look at different ascertainments for depression. We found it didn't really matter, but ideally stick to best practice, please. Anyway. And this is a paper we showed recently all about how what we basically found was that head injury associated with depression differently depending on the different types of ascertainments that you used for depression. So it can matter. Neuroticism, that's another very interesting one. This is kind of the only aspect of personality that Biobank have, really, and this is 12 questions which are collated to construct a commonly used neuroticism inventory, and that's in everybody at baseline. So you can perhaps begin to see that if you have a particular area, so if you're a personality psychologist, if you're whatever discipline you're in, there's probably some use Biobank has for the stuff that you do. So if you're a personality psychologist, you can look and see, well, do more neurotic people die earlier? Are their diets different? To what extent does cognitive health offset a link between neuroticism and death, et cetera, et cetera? So there's a lot of physical data in Biobank. There's also a huge amount of kind of personality and individual differences, which is quite interesting. So moving on to the imaging a little bit. So there's a little bit, this isn't on the ILOs, don't worry, of a sense that for a while their Biobank was becoming a little bit passé, and the reason was, I think, the same as often happens in many fields, is that there's a new toy, people exhaust that new toy, and it becomes a bit passé. I think with more and more time, the things that Biobank are doing and adding to the cohort become just incredible. And a big example of that is imaging. So imaging, Biobank is unlike anything in the world put together for brain imaging. So it's 100,000 people, I'm not one of them, who between 2014 to date, actually only a couple of months ago, they had their 100,000th participant, was in the news, which is unlike any other brain imaging or heart imaging or any kind of imaging you choose to suggest in the world put together. So, decades ago, a study could get into Nature with, 50 people from an MRI study, and now that, we're publishing 20,000, 30,000, 40,000, 50,000. It's really just absurd. This is described in the pretty dry, frankly, open access MRI protocol, which I encourage you to read with a glass of wine. And they did repeat cognitive assessments at the same time. So you have, the baseline cognitive data, and you have concurrent cognitive data taken at the time of imaging assessments. And they did various, I don't know if any of you care, they have various different sequences for the brain imaging stuff. Not all that you might ideally like, but they have some really cutting edge, amazing brain imaging pipelines, and you can look at various aspects of brain health, whether that's your hippocampus, which is a big memory center, total brain volume. You can look at white matter tracts, which are kind of the connective pathways between the brain, all sorts of stuff. It's really incredible. So the paper describes it. So very fundamentally with the imaging, whether you're interested in cardiac images, the heart, liver, kidney, heart, brain, whatever, the two fundamental routes are there are derived phenotypes which you can load up into Stata or R tomorrow that they have processed for you in Oxford, or you can go into the raw scans yourself, and maybe you have a fancy pipeline that you would like to enact on these raw scans. That can be really good. It can be a kind of unique selling point if you have the expertise and the time. A downside of the bulk scans is they are a faff to store. We at Glasgow fortunately have a specific exemption that we are allowed to keep the raw scans just now because we have specific things that we're doing to them that can't be done online. But the raw scans, it's a huge amount of storage, it's a huge amount of expertise and a huge amount of time for your pipeline that you want to enact upon these brain scans or heart scans or whatever. But it has to really be worth the effort to do that versus what Biobank have already done for us and is there and can be analysed tomorrow. There's a paper from a few years ago where we looked at, we essentially showed that people with heightened genetic risk for Alzheimer's disease who did not have Alzheimer's disease themselves showed differences in brain structure. And more laterally, we have started to look at atrophy. So there's 100,000 people with brain scan data, and they're bringing as many of those 100,000 back to scan again. Because a thing about cognitive aging is it's not really about the cross section; it's about the trajectory. It's about how your brain ages rather than its peak level. So we now have about 5,000 people with repeat scan data, which is again absurd. And we're starting to look at what actually influences the trajectories of brain atrophy across time. So genetics. We've kind of moved away from genetics a wee bit in SHW, I would say a little. Biobank, it's 500,000 people with whole genome sequencing. Again, another absolutely absurd thing. We've tended to look at the ways you can look at genetics at Biobank are you can either do a genome-wide association study, where you start with your trait and you're interested in what genetics contribute to that trait, or you can look and treat genetics as kind of like a proxy for variation in some pathway that you're interested in. So, for example, we were interested in, we're all familiar with GLP-1s, Ozempic, all that kind of stuff. We used genetic proxies of that to test whether or not they're associated with, whether use of that kind of drug might be associated with improvements in brain health, and the answer was a bit. On the other hand, like I say, this study from about 10 years ago now, was the first study in the world to show a biological substrate to personality, which is quite cool. So the various ways that geneticists could use Biobank are genetics, whether it's polygenic risk scores for things or rare mutations and how they associate with outcomes. People are interested in how does genetics influence how people respond to treatment. Can we cluster people according to genetic risk for things? Can genetics inform whether or not existing drugs could be repurposed for different pathologies? And can we use genetics, particularly GWAS, to identify drug targets that we've missed altogether? Because the vast majority of successful drug trials have evidence in genetics. Worry about that. Genetic data in Biobank has slightly gone away from us in Glasgow because we've probably got less bioinformatics support than we did a few years ago. A number of years ago, we had someone whose specific job was bioinformatics in Biobank. Also, as things have moved away from local storage, eventually all Biobank will be online. That would be very, very expensive to do high throughput genetic stuff in Biobank. So I don't know quite what we're going to do about that stuff in the future. Yeah. So what have we covered so far? Biobank is 500,000 people with various reassessments at varying sample sizes. They have NHS linkage until death. Biobank is a massive step up for sample size, occasionally at the expense of phenotypic depth. There's huge potential for cognitive, psychological, imaging, and really any kind of high throughput bioinformatics that you might be interested in. So I'll talk a bit more about the bioinformatics stuff later on. Now for some limitations about Biobank. So very fundamentally, there's a significant amount of selection bias in Biobank. And some people think that identifying, understanding, and correcting for this selection bias is the most important epidemiological question of our age, because it influences everything. We have attrition bias, which is sometimes participants have maybe, when you're looking at an exposure and an outcome, maybe they have died in particular ways which can influence estimates and can make something seem significant when actually it's kind of spurious. There are a significant number of spouses in Biobank that we can't really legally identify. Some studies have taken a punt at it by localizing where two people live, but officially you can't identify the spouses. To me, that's a pretty interesting bias, that there are various spouses in Biobank and we don't know who they are, whatever. And in that context, the reason it's interesting is because you have assortative mating, which is that on average, you are drawn to your spouse because they're on average similar-ish to you. Lag between assessments. Sometimes maybe you have variable A and variable B, but they're taken at completely different time points, which can obviously be quite problematic for causal inference. There are also issues with ascertainment, and what that means is that there are some aspects of Biobank which don't tally, which really kind of should. I'll talk to you about that in a second. So first of all, Biobank had a 5% response rate. That is, of the 500,000 people that attended, that represents 5% of the people that were invited in the first place. And it's very clear that they are not, your average bear. They tend to be more highly educated. They are lower than average deprivation. They're probably quite healthy and bright on average. And not only that, but subsequent to that, we have seen that it's very hard to show in detail that they are above average on everything when they first attended. But what we can do is we can show that the people who kept coming back are smarter and healthier and yada, yada, yada. And what we show here is that of the first 500,000 who attended, 15, 20 years ago, the ones who came back with brain scanning 10 years later were the ones who were at baseline tended to be better educated, they were younger, they were less deprived, lower risk of various conditions, just they were healthier in essentially every way, which has influence for what we then think we know about brain health particularly, or really any kind of health that is derived from the brain, the scan visits. And in that paper, we show that if you only included the people who subsequently attended the imaging, you would underestimate the negative effects of various cardiometabolic conditions on cognitive health. So it's very clearly showing that estimates differ because of bias in that paper. Similarly, what we also showed was that there is bias in who attended Biobank in the first place 20 years ago. There's bias in who attended the brain imaging and really any subsequent assessment. And there is also bias in who ended up with usable data. And so what we showed was that across a range of different variables, the healthier, the brighter, the yada, 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 were much more likely to move their head less in the brain scanner, because you want to keep your head still so they can get a good quality image. And they were also then more likely to have data that was complete, that had all the phenotypes that the scanner people wanted, and that could actually be used by researchers. So what this shows is that there's bias in who attends, right? There's bias in who kept on attending, and then there's bias in who actually provides data than that. So there's bias really throughout. I attended, Biobank had a wee party about 3 months ago, and that's where I got this lovely lanyard. And they had on the people who actually run Biobank. They were saying that there are a lot of spouses, and there were, I like this, there were a lot of Bentleys and Rolls Royces in that imaging car park, which when you were extrapolating to the general population at large, how many, how many patients that, you're doing research to inform their treatment, how many are going back to Rolls Royces in the car park? So I thought, anyway. As I said, there is evidence for assortative mating. There's also some really nice evidence in Biobank from Rebecca Rickman, who I hope you all attended her Morris block a few months ago, where they're interested in does in terms of assortative mating, does the behavior and tendencies of your spouse increasingly influence your behavior, your health? Which is why I think it matters quite a lot that there's actually quite a lot of spouses in Biobank. There's also again, huge trick for someone if they're inclined. There's a load of identical twins in Biobank, which nobody really touches. Everyone just excludes them because they're identical twins and they don't want the same person twice, effectively. No offense to identical twins, but it's an incredible resource. There's identical, like there's various behavior genetics approaches you can do to identical twins to estimate heritability of different traits, and they're just excluded. They're just chucked out the study altogether. So there's a win for somebody. Limitations, like I said, lag. So maybe I'm interested in this variable, and then the other variable you're interested in was here, or the other way around. It's hard to then say, well, we don't know what happened in the 5-6 years between these two variables, but we'll just let it fly. An additional point that someone, Jim Lucy, made when I presented something in about 2019 was, we're really getting far away from the baseline world that this came from, really. It pains me to admit it, but it's not 2006 anymore. Quite hard. And the world that people lived in, so things like pollution levels or smoking in pubs, yada yada. Maybe not 2006, but various things were really quite different about the world in 2006 compared to 2025. So actually, the things we can kind of say about biobank participants then is kind of becoming slightly less relevant as time marches on. Yep. This is a really interesting one. I said earlier on, you really need to look at different types of ascertainment in biobanks, so self-report, health records and stuff. The problem is that they really often don't correlate very well at all. And often when people are asked the same questions twice, they will give different answers. So this is a really, really fun paper. Papers aren't meant to be fun, but this one is. And essentially what they show is that, that there are predictors of giving inconsistent answers. Predictably or otherwise, it's men. Men are far worse at giving inconsistent answers between 2 time points, and which obviously has fundamental implications about the research you do in Biobank if you can't be sure that the answer they give on a Monday is the answer they give on a Wednesday. A few years ago, we looked at head injury, and there was essentially zero overlap between someone saying, I have had a head injury, and their health records saying they had a head injury. And that influenced, obviously, the estimates that we found when we correlated these things with brain structure and cognitive health. This is, as if you plan to use Biobank and you use it more, you will start to come up with your own favorite variable. My personal favorite variable is type of special diet. Please don't read ahead and spoil it. This field indicates whether a participant routinely followed a special diet. This data is an amalgamation of 4 yes/no answers. Some of the data regarding special diets is inconsistent with the actual food intake reported on the same occasion. For instance, around 1/10 of people indicated they followed a vegan diet, but also answered they had eaten meat the previous day. Stuff like that. So like I said, self-reported head injury doesn't tally with ICD codes. Another paper we did a few years ago, rheumatoid factor should be higher for people with rheumatoid arthritis, and it's not when you look at biomarker data. Similar medication use, they should be on chronic medication, and that doesn't tally well with people self-reporting their conditions. There are various, believe it or not, there are far more mundane minutiae about the cognitive data I could have bored you about, but I won't. But the cognitive data does have various flaws at baseline, which are really not ideal. So how do you actually go about getting the data? This is a really unfortunate time to get into biobank data, because for years it was local. It was on the J drive. You could register and get it. But increasingly, things are going to have to go online. A big motivation for that, from Biobank's perspective, is that they want more of it online, because it adds a layer of security, which means the GPs are more likely to agree to primary care data being shared. What you have to do is these 3 courses. You log into Biobank, what's called the AMS. So just type into Google Biobank AMS, and then you log into that. Then you do these 3 courses, which are a FAF, but very important. And then once you've done them, you can register with the Biobank DNA Nexus analysis platform. Within that, you can be put on one of, maybe you've got a Biobank application that a collaborator has. We have a good few in SHW just now. Mine happens to be called 17689, and it's all to do with the brain. And within that, you can see it's really not quite like Stata or R. It's its own very specific way of doing things. And what you would do is you would go down and pick the variables that you're interested in and add them to this tile. Then it will spit it out for you, and it'll just be ID number and these variables. And then it will save it as a funny kind of Biobank specific file in their own weird system. And then you would translate that into a CSV file, and then open it. I would suggest in R, really. I think you can do Stata, but you have to kind of make Stata work in quite roundabout ways, which is a faff. So I'd probably suggest R at this point. Be careful because but it is costed. And what that means is exactly as bleak as it sounds. You have to have either put money in to a big wallet, or you've got gang together with a load of collaborators and put a bulk amount down that you take it from, and then your analysis will be costed. And essentially that means as you do a regression, it will charge you a penny, that kind of stuff. For really high throughput stuff, I've been told that this can be about £300 per GWAS, which is £300 that we didn't have to find 5 years ago. There's no upbeat to that. So what have we covered? Definitions of things. Data. So the fundamental definition of Biobank and the structure, a bit about the data showcase, some of the data, some common limitations, and the analysis platform. The thing about the analysis platform is that we know it is going to change significantly between now and about springtime. So we know they're going to overhaul the whole thing. So to some extent, it might not be the smartest use of time to learn how to use it, because it is hard if they're going to upend it in 6 months anyway. But that's an individual call. What have we not covered? And the answer is a lot. So there's proteomics data that came from the baseline, accelerometry, that's things like sleep quality, sleep efficiency, physical activity, metabolomics, whole genome sequencing, identical twins. What's Biobank great for? The real nub of things. It is, and I think the sweet, if you think of like, an X and Y axis of phenotypic depth versus sample size, Biobank's slap bang in the middle. It's perfect. It's got a really big sample size, but it's got really nice phenotyping, largely speaking. It has about 15, 16 years of follow-up at this point. So when I first started using Biobank, we couldn't really look at dementia. Now we really can. It's really coming on song for dementia stuff, I think. There's some incredibly novel markers, so things like amyloid, which for only until very recently, you had to essentially, puncture somebody in the spine with their permission, ideally, to get amyloid, and now that's in blood, and it's really absurd. It can be very useful for if you're specifically interested in it as an area, in how does people's reporting of health differ with their health records. I think Biobank is where it's at, and I think that's a really nice area, is understanding discrepancies between what people say they have and what the health records say. I think that's really interesting. And as I've said, brain imaging is a generational step forward. So there really are some quite absurdly impressive things about Biobank. What is Biobank not great for? Routine real-world associations. So if you're just interested in, does disease X increase risk of disease Y, Biobank's probably not the best for you, and something like CPRD or Safe Haven's probably better. Cognition and personality is all right, and I think that's the case with many things in Biobank, where because it was a one-day assessment, the phenotyping is quite good, but it's not absolutely gold standard. It's also not amazing at modeling 2025 or the world going forward necessarily. Completely my opinion, take it or leave it. What do I think people don't do enough with Biobank? Does cognitive health buffer or predict later outcomes? Brain imaging, people have not used brain imaging enough, given how amazing it is, particularly functional MRI. Understanding the role of selection attrition bias, because it influences everything, and understanding how self-report and electronic health records complement one another. Here are various links to get you started if you want to use the data. I'll check out the Padlet if there's any questions. There's not. Grant, and here's various papers on if you want to get started on depression, bias. Kathy Sudlow paper there, which just describes Biobank, is quite good. Various cardiometabolic disease papers, brain imaging, aging, all sorts of bits and bobs. So yeah, I hope that's given you a rapid fire view of the fundamentals of Biobank. Do you want to talk now, and we'll do questions?
Yeah, we'll do questions. Yeah, that is yes. Please note, I'm not going to say very much because Donald has covered everything. Pretty much. I think I'm just going to more talk. Oh. I'm not as funny as Donald, so I'll not talk too long.
Go back.
Okay, so again, I don't need to go into this detail because Donald's covered all of this, and these slides as well have got all the links on here. So I guess this is just generally talking about, Donald's covered this really, but the different types of healthcare records that you might link to, different types of environmental data that you might be able to link to. And again, all of this is on there. So it's about basically going on the UK Biobank website and having a nosy around, because we can't possibly cover everything just now. I know there's lots of people in this room and probably online as well who actually know Biobank as well as Donald and I perhaps, or and have been using it in different ways. So again, everything isn't all in one place. So this just sort of highlights there's not kind of one place of Biobank where you can find out everything, because it's all kind of like round little corners and so on. So this just highlights things that I think are missing from this that are actually really important and could be useful in School of Health and Wellbeing. So within SHW, we've been using the data, what's about 2013, I think, across various projects, but we are very lucky to have a couple of really large projects that are really broad in what they can do, and what we have permission to do within Biobank. So this is the one that we've kind of held since 2015 within primary care. Again, mainly focused on multimorbidity, but treatment burden, comorbidity, long-term conditions, pain, frailty, social isolation and loneliness, physical activity, lifestyle. So a lot of those kind of more general demographic and health related factors related to outcomes, so particular outcomes we've looked at around hospitalizations, mortality, primary care. We have now had to move everything onto RAP because we've just recently had a renewal, and so anything that was getting renewed within Biobank is now having to go onto the RAP, which is this system that Donald was just talking about, so the online platform for analysis. So we're very much in the learning curves of that at the minute, and we do have some people that started moving on to that. And that's just like we must have had, I don't know, 30 odd publications out of this project now. But again, the reason we're lucky to have these big projects is that we can then have smaller projects within that. Biobank would be expensive to use if everybody had to apply for their individual project, which some people still do have to, but we've been lucky to see if to have these umbrella type projects. The other projects held by Rona, another big project, that covers a whole program of work, again, particularly around cardiometabolic, inflammatory, immune and brain related health outcomes, and does a lot around collaborations with School of Cardio and Metabolic Health as well. And then you kind of do bring in the genomic and proteomic data, and brain scan data, which again is something that we don't have in the other project. That kind of gives you ideas of again, loads of papers coming out of that, and that's a bit of an amalgamation as well of other historic projects within School of Health and Wellbeing that we had, and that kind of come together under this one. And part of that is to stop renewal of individual projects. Again, it saves costs. So as I say, a lot of these are long established projects that every 3 years we then have to renew. So it's important, I guess, if you're thinking about using Biobank, that you do factor in some funding. So that's funding to contribute to renewal of existing projects. Donald's mentioned the code for his project as well. So there's other projects around too. They are suitable for PGT and PGR projects. Obviously, PGT, we've got the problem at the minute with things moving on to this online analysis platform, where there's less experience overall in terms of supervision of that just now until we all get upskilled on that. The fees that we pay for renewal are dependent on the tier or the level of data that we've got. So for example, Rona's project is more expensive to renew than the first one that I showed you that we hold in primary care because of having that genomic and proteomic data and so on. But the key thing as well, which should be on this slide, is actually the expense now of using the online platform. So you do have to be able to pay for analysis. It's not insanely expensive unless you're doing the real high throughput stuff, as Dawn mentioned. And again, it's just about exploring the website, speaking to users of Biobank. So whether that be someone like Donald myself, but loads of PhD students and loads of other projects going on that have been using it as well. consider whether your work falls under some of the existing projects that are around, because that's how you can then add, it can be added value as opposed to then having to pay for a whole project yourself, and even just the review and the application process. Speak to us if you think your project falls under our remit, and then you have to register as a Biobank user on the website. CV, always use your University of Glasgow address. If you've got a different one, because you're maybe affiliated with another institution, if you want to be part of a University of Glasgow project, please use a University of Glasgow email address. Again, that's to do with funding, models, and so on. And then that's not about a new project, it's just about registering as a user. And then you can be added to the existing projects, and then data access can be granted. That's really all I was going to say, which was just additional to what Donald was covering anyway. So I think it's fair.
Yeah, we've got 10 minutes for questions. So.
Yeah. If you've got questions online, stick them in the chat or.
Yeah, just gonna ask about this. I just want to understand the cost thing. So how does it work? Like presumably whoever's the kind of integrally umbrella project would be the person that was kind of putting money into your account.
Yeah.
And then if a master student went on and started running loads of analysis with that.
No, you have to be, you're linked to different wallets, aren't you, in RAP? So everybody can have their own individual wallets, or then you can have a project wallet.
Yeah.
So
yeah. And is that where do funds come from within the school for people want to do that stuff? How does it all work?
So as Dawn said, this is kind of something new that we're coming to in terms of having to pay for individual analysis. It used to be we could just do whatever, we had the data and we could do whatever we wanted. Whereas now there's different models. It's about, you need to cost it into grants if you're applying, you know, if this is part of a grant or part of a PhD project, out of bench fees and research costs. Otherwise, we're finding bits and pieces of money here and there at the minute to cover it.
The answer is laboriously.
Yeah.
It's tricky. I think it's not just localized to Glasgow. I think huge. I think the landscape is going. How do we deal with this?
Yeah. So an important thing to say is actually early career researchers can get a pot of money put on there through Biobank, and there's a way to apply. But I think there's also pros and cons for some of that. And so I think initially everybody got £40. But now I think like early career, like postdocs and PhD students can get about £250 to £500, something like that. So everybody should, if you're using UK Biobank, you should be applying for that. I know a couple of people in primary care have done that recently. Hopefully I can circulate that information with the kind of that goes wherever Kevin puts the slides and stuff. But in terms of the 3-year renewal of these projects, that money tends to come from PhD students are using it, maybe comes from their research costs, other money within the school to try and renew them, because they're a great resource and they've you know, that are kind of widely open for others. Nowhere near as expensive as CPRD, like nowhere near as expensive in terms of institutional license, but still it's still money to find. Sorry.
Yeah, just curious about we talk about the limitations of the funding. So I was curious to know your perspective. Findings that researchers are doing. So you think there are, and there maybe certain areas where the accuracy and the validity of internal validity of the data is really good enough to make something out of the findings compared to other studies that use self-reports, which we are generally not as reliable and ways of collecting data.
Yeah, so it's often with Biobank, I think a lot of the time estimates are probably an underestimate of things. They're probably on the conservative side of things, whether that's through attrition bias or selection bias in the first place. So, for example, we know that at baseline there were below average rates of certain cardiometabolic diseases, for example, because it's probably the healthier people that are getting to the assessment centre. A very nice one I saw years ago was that there is a study did widespread genetic analysis of things, and they observed there were very, very, very few people with leg fractures. And then someone pointed out, well, yeah, it's probably because a lot of the assessment centres are upstairs without lifts. So there's all sorts of dumb little minutiae like that, which means that there are biases to people getting into the study. So it means that you're sort of restricting your range towards the healthier people, which almost by definition only means that you're going to slightly minimize magnitudes of effects of things. So whatever we find in Biobank is probably the thin end of the wedge a little bit. And that's where things like routine data sets like CPRD can be, they will always have a sort of a dovetailing benefit to looking at different data sets. It's a very, I mean, it's probably the fundamental limitation to Biobank, I would say, the idea. And I think correcting for it is if you can make a really good go of that, I think it's profound to what everybody does, is adjusting for bias in Biobank. Bias bank.
I think Donald has very nicely written everybody's strengths and limitations section of his of any papers in your presentation. It's really
it's not hard.
It's
I think the thing I would encourage folk to do is, if you're interested, the RAP, we need people to just have a goal, very, very scientific and conscientious, but just muck about with RAP, please, because we need people to understand it better and to lead.
So that we can share those skills. And I guess that's kind of what we're within team, the data and AI theme, the teams, that we can actually share that knowledge. Even if you're working on completely different projects, there's be a lot of like transferability of the skills and the knowledge that we learn, because there's no point us all working separately on these things.
RAP is a massive faff, but together we can make it less of a faff.
Jocelyn?
So if we say once we get students up and running, and their analysis move from student project to student project, so that can you share data across RAPs?
It makes that easier.
Yeah, that's
Yeah.
You don't have to lose that analysis time and process.
it can be copied between, as long as the person's an admin or is part of the different projects, that person can copy data between projects.
For better or worse, everyone can see everything on RAP. Like you go in and it's just like it's kind of like HPC, I guess, where I go in and I can see everything. I can see that you're running a regression, or I can see that someone's doing a t-test, and I can see everyone's folders. It's like Safe Haven, basically. You can see everything. It's like
on your project. If everybody's on your project, you can
you can see other people's folders and what they're all doing and all that kind of stuff. So it's all very.
And you can then access, the other plus points, I guess, instead of this being so negative alongside that. But no, but as in that's positive. But another positive is actually then you can constantly get the most up-to-date data, so that most up-to-date health, most up-to-date deaths and so on are all there for you. Whereas previously we were having it was quite a laborious effort to go in and download those with special access keys and so on. There are plus points to it.
So Heather has asked, going from complete novice to conducting analysis, how long would it take to learn? Do you want to take a go? Or
no.
So I think this really strongly depends on your question. I think when I have supervised students, it's tended to be, what's your exposure, what's your covariate model, and what's your outcome, and that tends to be whatever, not a huge number of things. If that's the case, I think it's quite easy actually to go and RAP and just go click, click, click. Here's my 10 variables. And then you put them into R. But that really will depend on what you're doing and what you want to do to those variables.
And in some ways, it'll be easier for new people, I think, than some of us who have been a bit set in our ways and how we've been using it for years and had the variables we want to use just right there for us, click of a button and so on. Whereas I think if it's all you know, and you learn that way, I think it'll be a bit more straightforward for you, to be honest.
They've changed the variable names on the RAP from what they used to be, which is a really sensible move. 15 years too late, where I have my scripts in the format they were, and they've made them something slightly more sensible now. So you've kind of lost all that build up. But if you're coming at it completely fresh, you might be like, what's Don's problem? Really?
But you can upload files. So we have uploaded some files to the RAP, some of our using files, if you like, that we've worked on. But
So we've got 3 more minutes. So I'd just like to say thanks Dom and Barbara for those presentations. Really
Oh,
informative for me, certainly, and sure everyone else. And in the last 3 minutes, so the last seminar we had, so CPRD has been spoken about a little bit. Last seminar we had was about CPRD, and at that time, the multi-study license for CPRD was going to run out in November. It's just to let this group know that that's now been renewed for another year. So if you're thinking about doing studies, CPRD was mentioned a bit, and are thinking that you might work on CPRD, then please email me and I can get you added to the Teams group for CPRD. So that's just a plug for CPRD at the end of the Biobank.
I think CPRD and Biobank complement each other really nicely. Completely different in the way they do things.
yeah, so studies that actually, you know, in a PhD or whatever, where you can do similar analysis on the two different things, but using different types of data can really quite strengthen work. So.
All right, great. Thanks a lot. Thanks everyone.
Okay.